So the facilitated diffusion of ions shown here can be seen as the cytoplasm on side and extracellular fluid. We have a high concentration that is now moving those molecules to the low concentration. We have extracellular fluid on one end and the cytoplasm on the other, where the cytoplasmic molecules and macromolecules are going to the extracellular fluid spaces. These carrier proteins can help transport both ions and other solutes, such as some sugars and amino acids as well. This movement is via diffusion and requires a concentration difference across the membrane and must bind to molecules as they transport them. So the saturation is important because it is the rate of transport that, limited by a number of transporters, allows for this change in the rate of this transport happens. So remember, the rate of transport is limited to saturation. When things are overly saturated or when things are in high concentration, remember they're going to move to the lower concentration, as we see in this image where it's moving to the inside of the cell where there is lower concentrations or saturation. Now, the facilitated diffusion by carrier proteins can be shown here in another image where we're looking at the extracellular fluid, these compounds and receptors binding to the proteins, allowing for different molecules to then open within the channel and send one through at a time. Within osmosis, our osmosis is defined as the net diffusion of water across the membrane towards a higher solute concentration. And cytoplasm of the cell is an aqueous solution that allows for osmosis to go on, where water is the solvent. And from here, we have dissolved substances that are solutes within these solvents. And in our image, we can tell how these different solvents move within our different water solutions. So semi-permeable membrane is shown here, where water is able to move freely. And we have a concentration of sugar shown on the left and diluted sugar on the right. Remember that things are going to move to the lower concentration, right? We're going to want to need to move towards a form of evenness based off of the components. But in osmosis, we see here that there's more water that is being drawn into the concentration solution because they are more concentrated. So more materials are able to bind to those water molecules compared to the other side where we have a lower concentration of sugar molecules. Within osmosis in the U-tube, we can see that urea molecules are important, right? Because they're going to be an organic molecule that binds these water molecules that are semi-permeable. The osmotic concentrations keep us going with this idea of osmosis. And when two solutions have different osmotic concentrations, the hypertonic solution equals the higher solute concentration, and a hypotonic solution equals the lower solute concentration. When two solutions have the same osmotic concentration, the solutions are then considered isotonic. As shown here in the image, we have the isotonic solution where there's a nice balance, homeostasis of water balance going on within these blood cells. We have aquaporins and specialized channels for water in the cell membrane that help facilitate osmosis across these different membranes, from the hypertonic being where the water is being shrunk and pulled out of the cell, and the hypotonic where the water is then H2O being pulled into the cell. With osmotic pressure, these forces are needed to stop osmotic flow. These pressures then allow for cells in a hypotonic solution to gain water, causing cell to swell and creates pressure. And cell walls can reach balance of the osmotic pressure, driving water in, with hydrostatic pressure, driving water out. The prokaryotes, fungi, plants, and many protists are seen doing this and understanding that water movement is based off of the effects of osmosis on these cells. So plasma membrane is not as strong and it may burst depending on how much fluid is going in and out of the cell, right? So if we have a hypotonic cell, depending on the strength of that plasma membrane, it is able to burst. And animal cells must be in isotonic environments, right? So these balanced homeostatic environments for things to operate accordingly. Within a normal red blood cell, we see that isotonic environment. And then with our low water concentration outside, we see that we're going to have a shrinking of these blood cells, right? So water moves out of the cell during osmosis, and we're going to see water move into the cell when there's a high water concentration outside the cell, which is where during osmosis, water moves from into the cell. Solutes and osmotic pressure in cells can also be seen in this image, not just with our blood cells we just talked about, but plant cells. So water is involved in all living organisms, right? Where that's the component that makes things living. And we see the flaccid cell and the normal turgid cell shown here that lets you see the difference from the isotonic solution, right, of a normal flaccid cell compared to the hypotonic solution of a cell being full of water. And then the other spectrum being the hypertonic, where we're seeing less water in the vacuole and the shrinking of the cellular components inside. We know that there's a maintaining of osmotic balance that needs to occur within these cells, right? And not just in plants, but in fish. So some cells use extrusion in water, in which water is ejected through contractile vacuoles. And isomotic regulation involves keeping cells isotonic with their environment. Marine organisms adjust internal concentration to match seawater, and terrestrial animals circulate isotonic fluid. Plant cells use turgor pressure to push the cell membrane against the cell wall and keep the cell rigid. So in our image here, we're able to see the freshwater telos and the marine one. And we see that the diffusion through skin happens in both species, fresh and marine water fish. We see that there's something going on when it comes to food. They're both able to eat, but one is now eating not just food, but is intaking seawater as well, correct? So there's a lot of ions and different things in there. Now, they're also both having diffusion through their gills, and the differences lie in between the dilute urine that happens in fresh water, right? Compared to the concentrated urine happens in the marine water, seawater fish, due to all the components that are needed to filter out that water. We also have the active uptake in freshwater fish, as we have active secretion going on in marine fish of those ions that are coming in and out of these organisms. Within active transport, we need to use ATP, right? So this is the energy that is required, and ATP is used to directly or indirectly to fuel active transport. This moves substances from low to high concentration. It requires the use of highly selective carrier proteins. These carrier proteins happen in this active transport form across this membrane, where we see the diffusion into the cell, the facilitated diffusion as well, which are both part of passive transport. We have active transport, remember, where it needs ATP, a form of energy, to allow these macromolecules to go from outside to inside of the cell. With active transport, the carrier proteins used in an active transport include the uniporters that move one molecule at a time, the symporters that move two molecules in the same direction, antiporters that move two molecules in opposite directions, and we also know that these terms can also be used to describe the facilitated diffusion carriers. So once again, an image of this shows the symporter having both directions of those molecules, two at a time. The antiporter being in both directions. So one direction being symporter, but being two molecules, right? Moving two, same direction. Antiporter being moving two molecules in opposite direction. And the uniporter moves one molecule at a time. Within the sodium-potassium pump, we see that there is a direct use of the ATP for active transport. This uses an antiporter to move 3 sodium out of the cell and 2 potassium into the cell. This movement is against the concentration gradient, right? And ATP is used to change the conformation of the carrier protein. So that active transport happens even against concentration gradients based off of the ATP needed to open those channels, right? So affinity of the carrier protein for either sodium or potassium changes so ions can be carried across the membrane. The image here shows that sodium channel allowing for molecules in to the cytosol, and potassium channels allowing these macromolecules to the outside of the cell. Sodium and potassium pump function can be shown in this image as well, where we're able to see the binding and connecting of the ATP along with the movement of sodium and potassium, where protein returns to the original conformation with low affinity for potassium, and potassium diffuses away as ATP can bind to start the cycle again. Now we're going to continue the cycle to where we're moving particles in and out, where bounded ATP is used to phosphorylate the pump, shown here. As we come through, move those particles, and this conformation has higher affinity for potassium, extracellular potassium, that binds to the exposed cell. Now, this binding of the potassium causes this dephosphorylation of the protein, right? So now we can then move to the next aspect of the ATP process and start it all over again, where we start off at the ATP 3 sodium ions that bind to the pump. Within coupled transport, we see that there's uses for ATP that are indirectly used. This uses the energy that is released when a molecule moves by diffusion to supply energy to active transport of a different molecule. Now, the symporter is used to have this coupled transport go on. Remember, these are needing the different energy additions of ATP. Glucose, sodium, symporter captures the energy from sodium diffusion to move glucose against a concentration gradient. With coupled transport via membrane proteins, we see that the movement of things happens outside to inside the cell. Again, this is just another image showing you how the sodium potassium pump is binding and moving and using ATP and these phosphate bonds to help power these transmembrane proteins. Now, the coupled transport protein can be seen in this image where there's a coupling effect, right? Where the glucose also comes in along with the sodium to then move inside the cell. We also have bulk transport that happens, where endocytosis is the movement of substances into the cell, and phagocytosis is where cells take in particulate matter. The pinocytosis is where cell takes in only fluid, and the receptor-mediated endocytosis is where specific molecules are taken in after they bind to a receptor. This all happens where we have exocytosis, where the movement of substances out of the cell requires energy. And in our image, we see that phagocytosis, where it's eating or engulfing particles to bring in the cell, as exocytosis is taking items and vesicles out of the cell. Here's another image of our endocytosis, where we're able to zoom in and see how this plasma membrane is now allowing and engulfing this bacteria, bringing it into the cytoplasm of the cell. This is called phagocytosis. Now, pinocytosis is where we're going to see that plasma membrane also take in solutes into the plasma membrane, as shown in this SEM image as well. We also have receptor-mediated endocytosis, where the target molecules are on the outside, and they are needing to have the receptor protein that is found on this cell membrane. These then connect to the clathrin as well, and as they move, they create a nice coated vesicle for the cell to have. So the human genetic disease from familial hypercholesterolemia, or LDL, receptors lack that tail, so they are needed to fasten the clathrin coated pits, right? And as this happens, we see that it does not trigger the vesicle formation, and the cholesterol stays in the bloodstream of the affected individual. Now that's the issue, right? So accumulating in the body is these forms of plaques inside these arteries that then lead to heart attack, causing strokes as well. Exocytosis, to show another image shown here, is where we discharge these materials out of the cell and is used in plants to export cell wall material. This also is used in animals to secrete hormones, neurotransmitters, and digestive enzymes. With that being said, we have reached the end of our chapter 5. If you have any further questions, please feel free to email me, and we'll be discussing the next chapter soon. With that being said, have a good one and good luck studying.